Welcome to the Melbourne Panalytical Web Seminar Program. Today's title is Innovative Assays for Neurodegeneration Drug Discovery Programs, presented by Elise Malavasi, Principal Scientist on the Neuroscience team for Concept Life Sciences. Prior to joining CLS, Elise obtained a PhD in Cellular and Molecular Basis of Disease from the University of Edinburgh, followed by 7 years of postdoctoral experience in Cellular and Molecular Neuroscience at the University of Edinburgh. Her previous research focused on the molecular mechanisms underlying major psychiatric illness and exploited several in vitro models, including neural cell lines, primary CNS cultures, and iPSC-derived neurons. More recently, she developed a microfluidic-based in vitro myelination model to investigate the mechanisms of myelination in the PNS. She has extensive experience in the development of in vitro neural models, light microscopy, and image analysis. Please know that we encourage interaction during today's presentation, and we will have a Q&A session directly following. Please take a moment now to find the interactive Q&A and chat icons on the bottom right-hand corner of your screen. Feel free to submit a question at any time, and we'll answer them following the presentation. If we don't have time to answer all questions today, we will follow up with you through the email address you provided upon registration. Our final interactive feature that you'll see on the screen in just a moment is a poll. This is an effort to get to know our live audience a little bit better and to help our presenter to tailor this talk to your experience level. Elise, thanks again for putting this talk together. Could you give us a little insight into what we'll be seeing while people take the poll?
Yeah, of course. So first of all, welcome everyone, and thanks for joining us today. So the aim of my presentation today is to give you an insight on some of the types of assays that can be used to investigate the efficacy of novel drugs on key neurodegenerative processes, such as neuroinflammation and demyelination. So I will start by describing simpler cell-based assays and then move on to more complex ex vivo studies with a view to exemplify how the drug discovery process can be de-risked by assessing candidate drugs in increasingly complex and representative models. And finally, I will present a case study to exemplify how a cascade of increasingly complex assays can be used to test candidate drugs in the context of multiple sclerosis.
Thank you for that. If everyone is taking the poll could just wrap up really quick, then I can share the results with everyone. So everything is wrapping up now, so you'll be able to see the results of the poll on the screen. Elise, if there's anything that you want to summarize from that, feel free. It should pop up in about 5 seconds.
Yeah, seems like, there are some people with us today that are specifically interested in multiple sclerosis, which is what I'm going to be focusing on in the last part of my talk, but also some more interested in Alzheimer's and Parkinson's, which is also very interesting because obviously neuroinflammation also applies to these disease areas as well.
If you are all set, Elise, I will hand over the controls to you, and you can go ahead and begin your presentation.
Okay, so at CLS we have developed and optimized a series of in vitro and ex vivo assays to model neuroinflammatory and neurodegenerative processes and test novel drugs in these contexts. So these assays are largely based on primary rodent cells and tissues because these more closely recapitulate the in vivo biology of the CNS. So we start by isolating rodent brains, either mouse or rat, and from these brains we can either prepare slices and culture them in a dish, or we can dissociate the brain and isolate and culture individual CNS cell types, including astrocytes, microglia, neurons, and oligodendrocyte precursor cells. And in some particular applications for which primary rodent cells are not the best option, if, for instance, if it is crucial to use human cells, we can use cell lines instead, for instance, microglial cell line HMC3. or human-derived induced processed pluripotent stem cells. So once we have our tissue or cells in culture, you can use them to model a range of processes, including neuroinflammation, neuroregeneration, neuronal function, or toxicity. And so one of the areas we specialize in is neuroinflammation, because this is known to be a key player in neurodegeneration. And in the CNS, microglia and astrocytes are the primary effectors of neuroinflammation. And under physiological conditions, these cells can be found in the brain in a resting state, but when they are exposed to certain stimuli, such as an insult, which could be an injury or an infection, they become activated and they can polarize towards pro or anti-inflammatory phenotypes. And the function of this activation is to repair the damage or clear the infection and essentially repair and restore the tissue. But if the insult or damage is chronic, as in neurodegenerative diseases, this activation then becomes deleterious and can contribute to tissue damage itself. And we know that in neurodegeneration, the polarization balance of astrocytes and microglia is shifted towards damaging pro-inflammatory phenotypes. So cultured microglia and astrocytes can be stimulated in vitro to assume pro or anti-inflammatory phenotypes, and drugs can be tested for their ability to influence their polarization. So the outcome can be read out using a variety of approaches. So we can look at polarization marker and or target protein expression using multiplex immunostaining or flow cytometry or Western blotting. We can look at the same at the gene expression level using qPCR or RNA-seq, or we can look at profiling the cytokines that are expressed by these cells using immunoassays such as ELISA or multiplex ELISA, including MSD or Luminex. Or we can investigate activation of particular signaling pathways, for instance, using homogeneous time-resolved fluorescence or HDRF, which is a technique based on FRET. And for more functional outcomes, we can use live imaging of these cells via the InQSight system, which is essentially a live imaging system located inside an incubator. And this allows us to monitor and document processes such as phagocytosis, cell migration and proliferation, or apoptosis kinetically in real time without being limited by having to select a certain endpoint. So we can use two distinct paradigms to test candidate drugs in the context of neuroinflammation. We can test the drug's ability to inhibit the damaging pro-inflammatory polarization of microglia and astrocytes in a pro-inflammatory environment, which we mimic using LPS. And in this case, our positive control is dexamethasone, which is a very powerful anti-inflammatory drug. Or we can do the opposite. We can promote the anti-inflammatory polarization of naive microglia and astrocytes. And in this case, we use a polarization factor, IL-4, as a control. And so in these graphs, you can see an example of how selected polarization markers change at the gene expression level, which was quantified by qPCR in microglia after either a pro-inflammatory polarization on the left or anti-inflammatory polarization on the right. So we selected these two widely adopted markers of polarization, NOS2 and arginine 1. NOS2 is a marker of pro-inflammatory phenotypes, and ARG1 is the opposite, marker of anti-inflammatory polarization. And so you can see on the left that LPS stimulates an increase in expression of both, but in a pro-inflammatory environment, NOS2 vastly outcompetes ARG1. And the opposite is true in an anti-inflammatory environment stimulated by IL-4 treatment, where we see that in this case, the expression of ARG1 outcompetes that of NOS2. Here I'm showing an example of astrocytes, of microglia M1 pro-inflammatory activation, but this time using an imaging readout. So we quantified the percentage of iNOS positive cells. iNOS is the protein product of NOS2. And you can see that this increases upon stimulation with LPS, and this increase is then suppressed by dexamethasone. So in addition to looking at polarization markers such as ARG1 and iNOS, we can quantify gene expression changes in pro or anti-inflammatory cytokines. So in the example shown here, we quantified pro-inflammatory cytokines IL-6 and IL-1 beta, and anti-inflammatory cytokine IL-10. And on the top, you can see that upon LPS stimulation, you see a very big increase in expression of the two pro-inflammatory cytokines, but also of the anti-inflammatory cytokine. And dexamethasone can suppress expression of the pro-inflammatory cytokines, but further increases that of IL-10. And in an anti-inflammatory environment, we see that expression of all three cytokines is inhibited. And in addition to LPS, astrocytes pro-inflammatory polarization can be induced in vitro by treating naive cells with a cocktail of interleukin-1 alpha, TNF-alpha, and C1q. Which are all highly expressed by activating microglia. And this was reported by Little and colleagues a few years back. And so here in these graphs, again, we quantified the expression changes in these two genes, NOS2 and ARG1, both after stimulation with the cytokine cocktail and LPS. And you can see, as seen for microglia, in a pro-inflammatory stimulus produces a very large increase in expression of NOS2, which outcompetes that of ARG1. So as shown for microglia, using this stimulation paradigm, we can quantify cytokine gene expression changes. Here we looked at pro-inflammatory interleukin-1 beta and TNF-alpha. And we can see that both the cytokine cocktail and LPS can drive expression of these cytokines, whereas interleukin-4 suppresses their expression. And to complement gene expression profiling in activated astrocytes and microglia, we can also look at cytokine secretion by these cells using ELISA or multiplex ELISA. And this can help us build a very detailed picture of the inflammatory polarization of these cells and how it can be affected by a test substance. So recent studies have implicated the inflammasome in neurodegenerative diseases. Inflammasomes are multimeric protein complexes that assemble in the cytosol in response to infections or abnormal molecules derived from host proteins. And they induce the activation of caspase-1, which is a pro-death factor, and also promote further inflammation. So NLRP3 is one of the best studies and best studied and best characterized inflammasomes. It is known to require two stimuli to be activated. The first one, or priming stimulus, which in vitro is typically LPS, can induce expression of NLRP3 and also interleukin-1 beta. And a second activating stimuli leads to the formation of the actual NLRP3 protein complex. Activating stimuli can vary in their nature, but in the example depicted here, it is potassium efflux via ion channels, which is stimulated by ATP and nigericin. And so the inflammasome, once activated and assembled, then induces caspase-1 activation, and in turn conversion of pro-interleukin 1 beta to interleukin 1 beta, which is then secreted by the cell. Brain slices are an excellent model to study inflammasome activation, as they maintain all the relevant cells, the relevant structures and organization of the brain. In a typical assay, slices are obtained from neonate mice, and they are cultured in inserts in 6-well plates. Then they are exposed to inflammasome activating stimuli such as LPS and ATP in the presence or absence of a test substance, or the positive control MCC 950, which is a very potent NLRP3 inflammasome inhibitor. And then the secretion of interleukin 1 beta in the medium is quantified by ELISA. And so using this paradigm, we can also simultaneously quantify other markers, such as markers of pyroptosis, which is a type of rapid cell death induced by inflammasome activation, or other inflammasome effectors. Another major area of focus for us is neuroregeneration, and in particular the preservation and regeneration of the CNS myelinating cells, oligodendrocytes. And as I mentioned earlier, we can isolate and culture oligodendrocyte precursor cells from rodent brains. And in vitro, we can assess their proliferation and their differentiation into more mature myelin-producing oligodendrocytes using a defined differentiation time course. And in the example shown here in this image, I'm showing an example readout for this type of assay, where we stain our culture for a marker of mature myelin-producing oligodendrocytes, which is MVP, which is shown in red, and also quantify the number of immature OPCs using NG2 in green. So we can also look at expression levels of targets of interest in mature oligodendrocytes, again using staining or qPCR or both. So this graph shows an example of an OPC differentiation assay in which staining was used as a readout, and we quantified the change in the proportion of MBP expressing, so differentiated cells, upon stimulation with a positive control T3. And we typically observe a 2 to 4-fold increase compared to vehicle. Typically, during OPC differentiation, many genes are expressed transiently. So it can be very helpful to define the expression profile of the target of interest during OPC differentiation, so that the treatment window can be matched to the target expression window. And so in this example here, we quantified differentiation marker MBP and also a target of interest by both qPCR and staining. And as expected, as the differentiation progressed, we see more and more expression of MBP. And also in this case, the target gene was expressed, reached its peak expression around day 6. And we also confirmed that by staining. So while studying OPC differentiation and maturation in vitro can be very useful to reveal the remyelination promoting potential of candidate drugs, it does not recapitulate all of the complex cell-cell interactions that exist in vivo. But these can be modeled much more closely using organotypic brain slices to study remyelination. As illustrated here, we can produce brain slices from neonate mice. So in mice, myelination occurs largely postnatally. So these neonates have virtually no myelin in their brain. So we can culture these slices in these inserts in 6-well plates and allow for spontaneous developmental myelination. Once this has happened, we can then introduce an insult. For that, we use LPC, or lysophosphatidyl-choline, which is toxic to oligodendrocytes, and so essentially destroys the myelin. So after this demyelination, we can then add test compounds or a reference substance to essentially compare their ability to promote repair of myelin. And our typical readout in this case is immunohistochemistry, where we quantify the amount of myelin that has been produced. So here you see MBP, a myelin marker in red, and axon stained with neurofilament in green. And essentially we then quantify the proportion of axons that are covered by myelin. And we can also quantify any other myelin-associated or target genes by qPCR or RNA-seq, or use specialized histology techniques for more complex investigations. So our histology services allow us to assess expression of targets and validate their expression in not just animal models, but also human tissue, both diseased and control tissues that we can source through UK biobanks. And so our histologist can provide tissue processing, antibody and biomarker panel optimization, as well as protein and mRNA quantification in the tissue, fully automated tissue processing and image acquisition, as well as expert image analysis, which is all performed to GCP standards. And so we are constantly looking to expand our capability, and we are currently developing new assays. Given our focus on neurodegenerative diseases, we are focusing on validating primary neuronal cultures, starting from simpler monocultures and then progressing to more complex co-cultures with glia. So we will soon offer automated kinetic analysis of neurite outgrowth, including neurite length, neurite complexity, branch points, and quantification of neuronal cell bodies using our IncuCyte platform. So in the example shown here, you see a neuronal culture that was imaged live on consecutive days. And so this data acquisition was done without the addition of any dyes or viral vectors to deliver essentially labels to the neurons. Neurons are simply imaged by bright field, so with minimum disturbance. And this allows you to essentially monitor their growth and test the effect of substances with neuroregenerative potential. We can multiplex these readings with cell health measures. And then we can, at the end of the assay, we can analyze synaptic markers or any proteins of interest via staining, and also profile gene expressions via qPCR or RNA-seq. We can also offer assessment of neuronal and astrocytic activity using calcium imaging. So while my team and I in Edinburgh focus on neurobiology and neuroassays, we also have colleagues in England that specialize in computational and medicinal chemistry and offer a comprehensive array of drug discovery services, which include but are not limited to CNS drugs. And what I think could be of particular interest for you today are our CNS tailored artificial intelligence and chemoinformatics services, which include CNS multiparametric optimization and also machine learning algorithms that can reveal the potential of a candidate molecule to effectively cross the blood-brain barrier. So our chemistry team also offer over 13 in vitro ADME screening assays, as well as extensive in vitro toxicology profiling. But most importantly, for those of you who are interested in CNS drug discovery, they can assess binding of small molecules to brain tissue using rapid equilibrium dialysis of brain homogenates. And they can also offer a range of neuropharmacokinetics and bioanalysis assay that can reveal the ability of a molecule to distribute to the brain in a unbound and thus active form. So in the last part of my talk, or the next part of my talk rather, I will introduce multiple sclerosis. So multiple sclerosis, or MS, is an autoimmune disease of the CNS, and as such, it affects the brain, the spinal cord, and the optic nerve. Its most prominent pathological feature is the degeneration of myelin. It is a lifelong condition and can be very disabling, but symptoms vary greatly in both nature and intensity. It is the leading cause of non-traumatic neurological disability in young adults, and as such, it is a prominent societal issue. Its prevalence varies between 50 and 300 individuals per 100,000 people, and it is currently incurable, but therapies exist to alleviate the symptoms and slow the progression of disease. So although the etiology of MS is still unclear, we know that CNS inflammation is the primary cause of damage in MS, and chronic inflammation can destabilize the blood-brain barrier and allow activated lymphocytes to invade the CNS. And in the CNS, T cells stimulate both B cells and microglia, resulting in the release of myelin reactive antibodies and pro-inflammatory cytokines, which then cause damage to myelin and ultimately to neurons as well. And reduced glutamate uptake from activated astrocytes also leads to accumulation of glutamate in the extracellular space, which then causes excitotoxic damage to neurons, further exacerbating the damage. In the final part of my talk, I will exemplify how selected assays can be built in a cascade of increasing complexity to reveal the potential candidate drugs in the context of MS. So possible therapeutic strategies in MS can be aimed at reducing microglial inflammation by inhibiting the deleterious pro-inflammatory microglial phenotype, or by promoting the neuroprotective anti-inflammatory microglial phenotype. Or they can be aimed at promoting the clearance of damaged cells and myelin debris by microglia through either an enhancement of migration of microglia towards the lesion areas, or through an increase in microglial phagocytosis. Alternatively, they can target OPC biology by, for instance, increasing the differentiation and maturation of OPCs, or by stimulating their myelination and migration to lesion sites. So with this focus on microglia and OPCs in mind, the following cascade of assays can be implemented. So stage 1 assays are the least complex and the highest throughput, and then one can gradually progress to stage 5 assays, which are obviously the opposite. So for microglia in stage 1, we can look at activation and polarization using the assays that I've described earlier. And similarly for OPCs, we can look at differentiation and proliferation in vitro. And we can also look as a second stage, although stage 2 assays can also be performed in parallel to stage 1, because test substances can be active in one and not the other, or vice versa, or in both. So we can look at microglial migration and phagocytosis using, for instance, our live imaging equipment. So for migration, we can perform something called a scratch wound assay, where essentially microglia are allowed to grow as a monolayer, and then a physical scratch is produced in the well, and cells are observed and documented as they migrate towards the wound and repopulate that area of the well. For phagocytosis, we can monitor internalization of fluorescent particles, or we can use myelin debris. And so for OPCs, so once they are differentiated into oligodendrocytes, they acquire the ability to myelinate. But if they're cultured in isolation, they do not myelinate because they do not have anything to myelinate. So one possibility is to use an artificial substrate such as nanofibers, because in fact it was discovered, and it's now very well documented, that OPCs do not need axons to produce myelin. They can also myelinate inert artificial substrates. And so the use of nanofibers offers the advantage of providing a very standardized and more reproducible assay, because these nanofibers can have a very constant diameter. Obviously, their physical and chemical properties are fixed, and they can be regularly spaced and aligned in the dish, so that image acquisition and analysis is greatly simplified. So if a test substance proves successful in these simpler cell-based assays, we can perhaps progress them to more complex co-culture assays. So we can, for instance, culture neurons with OPCs and evaluate essentially the test substance ability to promote myelination of actual neurons. Or we can co-culture OPCs with microglia and evaluate, if, for instance, the effects of activated microglia on OPC myelination. So the next stage, we could be moving to even more complex ex vivo assays, such as the organotypic slice culture assays that I've illustrated earlier on during this presentation. And essentially, the final stage will be to progress to an in vivo model. And we are currently developing our cuprison remyelination model. So cuprison is very widely used to induce widespread demyelination in rodent models. So after cuprison treatment, mice can be administered test substances to evaluate their potential to promote repair of myelin. So this brings me to the conclusion of my presentation. And just as a final note, I would like to add that while we do have a range of validated, ready to use assays, we do understand that it's not possible to cover every scientific question with a simple set of assays. So we are always very keen to fully understand each individual project and to tailor the science around the question. And so if that means, for instance, developing new readouts or even developing completely novel assays, we're always very happy to work collaboratively with our clients to do so. And and finally, we do have a dedicated quality team to ensure that our data is not only delivered on time, but it's always thoroughly checked and accurate. I think that's all I wanted to talk about today. Thanks very much for listening. And I'll hand things over to Caitlin.
Thanks, Elise. So we'll now get started with our Q&A session. As a reminder, you can use the Q&A option in the bottom right-hand corner of your screen to submit your questions. If you're listening to this webinar on demand, please know that your questions are important too. You can simply email us at the address that's on your screen now, or you can visit our website and find local contact information there. At this time, you'll also see a second poll on your screen. Please take a few moments to respond to that as well. So our first question that we'll get started with is, can primary rodent cells be replaced with human iPSC-derived cells in the cell-based assays we described today?
Well, they certainly can. We are currently working with specialized suppliers of human-derived iPSCs to establish these cells in our labs, and in the very near future we're planning on essentially validating all of these cells in our assays. Just as a note, it's while astrocytes and neurons are, iPSC-derived astrocytes and neurons are quite widely available and can be sourced from many different suppliers. We haven't been able to find a reliable source of OPCs. So yeah, I would say definitely yes for astrocytes and neurons. It might require a bit longer to develop OPCs.
The second question we have, are these assays based on WT cells or tissues? And if so, is it possible to use disease models instead?
Yeah, so the assays I've described today are all based on wild type cells and tissues. However, we can absolutely use disease models instead. There's nothing really preventing us from doing so. It's again for protocols for isolating astrocytes and neurons. Obviously, we already isolate neurons from mice. For the isolation of OPCs, we currently use rats. So if someone wanted to study a mouse model, for instance, and wanted us to isolate mouse OPCs, that would require a bit of optimization because mouse OPCs are notoriously difficult to obtain and isolate. But for neurons, slice cultures, and astrocytes and microglia, that would be easy to do.
Next, we have, could you tell us which concentration you use to simulate astrocytes?
Unfortunately, I can't remember off the top of my head, but I will definitely check and get back to you.
Looks like that's all we have for now. If anyone thinks of any questions later, feel free to send us an email at events@malvernpanalytical.com. We can have Elise get back to you as soon as she gets a chance there. Elise, do you have anything else you'd like to add before we wrap up for the day?
Well, I just want to thank everyone for joining me today. I hope I was able to give you at least a high level of this understanding of the assays we can use to study candidate drugs in neurodegeneration. And if you would like to find out more about any of these assays that I discussed today and more, and perhaps how these could be tailored to your particular studies, please do get in touch with me. And thanks again.
Thank you, Elise. Have a great day, everyone.